an inventory of phytoplankton in the coastal waters of Sarangani Bay, particularly in the inside portion of Sarangani Bay, which is MSM or the Maasim Malapatan. This study is all about the inventory of phytoplankton in coastal waters of Sarangani Bay, wherein the phytoplankton nets is vertically towed into the water with a depth of 50 meters, 5 meters, and 100 meters, and collects samples in each depth, then placed in a opaque bottle that is properly labeled and stored in a cool ice box to avoid direct sunlight. Afterwards, it is viewed under the microscope and analyzed what phytoplankton are present in their area in relation to physical chemical parameters and their abundance and richness. This study's main objectives intended to conduct an inventory of phytoplankton in the coastal waters of Sarangani Bay. Specifically, this study aimed to, first, to describe spatial distribution of phytoplankton species, their composition, diversity, and abundance in Sarangani Bay. Second, to determine the physico-chemical parameters of the water in the study site. And third, to determine the effect of physico-chemical parameters on the phytoplankton composition, diversity, and abundance in Sarangani Bay. The materials needed in the conduction of phytoplankton are opaque bottles, phytoplankton nets with 25 microns mesh size, cooler with ice, 30% formalin and Lugo solution for preservation, GPS for location, pH meter, DO meter, and other apparatus to check temperature and conductivity of the water. I conduct my data collection at the Laboratory of College of Fisheries. The 1 ml samples per depth of 3 replicates are carefully put in a Sedgwick rafter slide and view under inverted microscope and record the data I gathered in a data sheet. Afterwards, I sensibly identify the phytoplankton samples using lots of manuals and identification guides. Then, the samples are grouped into genus or species level and count their cells manually. That's all. Thank you.